Well, I always love the creative titles the Farming Smarter folks always come up with for the various presentations. So next up, we're going to do a little crime solving as we have Syama's Inquest, Justice for Peas. Dr. Syama Chaterton joined Agriculture and Agri-Food Canada at the Lethbridge Research Centre back in 2011 as the pulse and special crops pathologist. Her research focuses on management of root and foliar diseases of pulse crops and on molecular diagnostics and identification of some of these soil-borne pathogens. And she was the first to identify that Aphanastes root rot was present here in the province and has been on a challenging journey of discovery of this troublesome pathogen ever since. She is chasing the dream of developing a robust decision support system to reduce root rot risk for a sustainable pulse production. So here today, she is going to explore the crime of root rot nutrient robbery and how to protect peas from this gang of invaders. Let's say hello to Dr. Syama.
Title. That was like a Friday at the end of the day. I think I maybe had one beer. I was like, okay, I got to make this fun. This was a really, really popular show in Vancouver in like the late 90s, Da Vinci's Inquest. I don't know if anyone remembers it. So I just kind of changed it to Syama's Inquest. And then when I went looking for a poster, I thought it was amazing how easily I could change it to fit what we were talking about. So in the underground of peas, every pathogen has a secret. I think it was originally in the underground of crime. Every criminal has a secret or something. Anyway, so it fit in really well with that title. So what we're going to start talking about is our victims of this crime. These are pulse crops, and in particular, peas and lentils. I think Ken just talked about why it's so important that we can include pulses and legumes in our rotations, particularly for these when we're talking about agriculture climate change solutions. We all know that pulses produce these beautiful nodules that reduce the use of fertilizer, or that you don't need fertilizer in your pulses, and then you also get that fertilizer advantage for the next crop. So really an essential part of sustainable agriculture and looking at climate change solutions. As well, they use less water than a lot of the other crops, have a smaller kind of footprint, and they also improve soil health. I know this is kind of a controversial topic, whether you believe it or not. The consumers believe it, and we really have this increased demand for pulse protein. And I think both, when you look at climate change and FAO, all of them recognize the importance of pulse protein in reducing climate change and replacing some of that meat protein with pulse protein. And so we can see that consumers are really jumping on board with this pulse protein. And I found this nice graph that was in Western Producer that really showed how consumers, how this demand for pulse protein has really been increasing. So in comes our gang of invaders, the root robbers, which actually fits also really well with the root rotters, so we could go either way with this one. And kind of our star player is Aphanomyces euteiches, but also featuring Fusarium avinaceum and Fusarium solani. There's very specific reasons why I chose the little emoticons that I did for each pathogen, and I'll make it clear as we talk about them. And then the other thing I wanted to include was this idea that there's other pathogens involved. We have Pythium, Fusarium, Rhizoctonia, and I gave them little happy faces because they're kind of like your little hapless criminals. They're maybe just along for the ride, but they don't really know what they're doing there. We don't really know what they're doing there, but we find them often in the system. Whereas Aphanomyces is really kind of the big bad, or the bully of the system, and then Fusarium avinaceum and Fusarium solani are like their henchmen. And it's brought to you by wet soil, crop history, and tight rotations. And I'll get into all of those different things throughout the presentation. So our big bad, you always have to have the big bad of your story, right? Here it's Aphanomyces euteiches. And this guy just kind of sits and waits in the soil and really wants to attack those peas and lentils that are in there. So it's sitting there waiting to put up a big fight. And what we see with this pathogen, why we call it our big bad, is because in really wet years, you can see that your pea fields can end up looking like this. We haven't had really bad disasters like this in the past few years because it's been drier, But if we're back into a wet cycle, it's something we have to look out for. And then when you pull out those roots, what you see is why we call them the root robbers, is there's really nothing left to that root system. They just kind of eat away at all the lateral roots, eat away at all those nodules, so you have nothing left there, and really you're just left with this little stump of a root. And if we look into kind of the life cycle of Aphanomyces and why it's our big bad, it's because it produces these spores called oospores that can sit in the soil and survive for a really long time. Towards the end of the presentation, we'll get into a little of the science behind that. But they sit there and wait, and you give them their recipe of nice juicy pea roots and water, and that's exactly what they want. And they produce these little, another cycle of these little cells, they're called zoospores, and basically they can swim around, find that root, infect it, and cause disease. And then once they've robbed all those roots of their nutrients, eaten up all the juicy nodules, everything that we like for those pea roots, then they have to go into survival mode, so they'll go back to produce those oospores. And then those oospores sit and lay in wait for the next time you have a pea or lentil crop. So really what we find is that these root robbers destroy roots. But if you notice in the last picture, I showed a really nice picture of what Aphanomyces root rot looks like. Honey brown caramelized roots. We go out into the field, we never see roots that look like that. We see roots that look like this. And this is why we're talking about kind of a gang of root rot invaders or root robbers, because they're all working together to really destroy the roots. And so most often what we see is that the roots are infected with this pathogen complex. So I'll talk a little bit about our two kind of prime players in the complex, one being Fusarium avinaceum. And I gave this guy kind of like a zombie chomper face because it doesn't care what it eats. It really will just eat anything that's there in the field. It has a very wide host range. We find it on almost all the pulse crops. We find it on cereal crops. We find it on canola crops. So it's just like, give me roots, and that's what it wants to do. So we did look a little closer into some of these pulse crop options because this has such a wide host range, to look at, okay, well, what pulses does it like the best? So we look both at emergence. So that's just, you can see the picture there where there's kind of a rotted seed. If it's present at seeding, it'll just rot the seed and the plant will never come out. So that's actually where seed treatments are good at protecting against Fusarium avinaceum. And then we can see that for the most part, we saw faba beans, and actually one variety of chickpea did very, very poorly with emergence with Fusarium avinaceum. Whereas peas and lentils, soybeans, some of the other chickpea varieties actually did quite all right. But I think just looking at that responsive chickpea, if anyone's grown chickpea, you know it's pretty much impossible to get establishment without a seed treatment. Very, very difficult for organic growers to get chickpea established, and that's because it's so sensitive to some of these Fusarium pathogens. When we look at disease severity, so this is more like, okay, so your plants came up, now what happens to the roots? We're looking at really, okay, what's that degree of rotting or robbing of your roots? And then we can see that faba bean and pea are the most sensitive to kind of the later stage root rot that's caused by Fusarium avinaceum. And like I said, it is associated with Fusarium head blight of cereals. We do find it in canola. Almost any crop that you go and look for, you will find Fusarium avinaceum. So just kind of this generalist, kind of like zombie-like pathogen that just wants to eat any crop it can get its hands on. When we look at Fusarium solani, this one I gave sort of a more like cunning persona. And this is because it's really focused on just a certain, it has a very specialized group of pulses that it likes. So when we look at emergence, we can see that everything did fairly well, except again, faba bean and another chickpea cultivar. But when we look at disease severity, you can see it really causes disease on faba bean and pea, and really we don't really see much disease at all on any of the other pulse crops. So you kind of have this interplay of all these different pathogens on these roots, some that cause more damage than others depending on what pulse crop you're looking at. So how do we stop this gang of root rot robbers? I mean, look at those peas. They're so cute, right? We want them to survive. And I did not know give peas a chance was such a big meme, but I went looking for it. Apparently it's a really big thing. And I guess then enter the mad scientist or the mad investigator. And I will say some of the things that we've tried are pretty crazy, but like Ken said, it's for us researchers to try things that fail rather than have farmers try it and fail. And really why we're searching for a solution is because what we talked about earlier, that disease plants, they no longer are providing that nitrogen benefit. Both, you're not getting very much yield out of your peas and you're not getting that benefit to your next crop. And another big question that we really don't know is whether there's actually an impact on the protein content of those seeds. So when we're looking at pulses being such an important part of consumer protein, plant protein now, if we are reducing nitrogen fixation in the roots, they're not picking up their nitrogen, what's happening with that protein content? And I think that's a question that really hasn't been looked at. So now I'm going to sort of switch over from the fun kind of criminal side and just get into the boring science side. I will try to make it as interesting as I can, but we will see. So when we look at management options, a lot of these are things that we have tried, and really the only thing that has worked is looking at crop rotation. And we look at crop rotation kind of under two different categories, and I'll get into those later, is one as a method of prevention, and then really it's a method of avoidance, and that's really the biggest tool that we have is avoidance. I think we are all very familiar with this idea of avoidance, quarantine, isolation after our 2 years of COVID, and honestly, that's almost the same thing that we're teaching for these root rots. So when we look at genetic resistance, really our peas and lentils have very little genetic resistance. We look at fungicides. There's a couple seed treatments registered for suppression, but it's only early season suppression. They don't last the full length of the growing season. We've done some work with biological controls. So far, nothing has really shown any promise. It's hard to get biological controls to work in the field just because of the conditions you need for good colonization. Soil amendments, there's some things that we're looking at. The problem is it's so much work to get them into the soil that even if it works, it might not be a feasible option in the long run. So we'll get into really talking about prevention and avoidance for the rest of this talk. So prevention is just really one easy slide. And if you don't have root rots, yay for you. So the question is, you don't have root rots, you don't want to get root rots, how do you prevent root rots from developing in your field? And the answer to everything, if you ask a pathologist, is rotation, rotation, rotation. So, if you're a new pulse grower, you're just interested in starting to include pulses in your farm, do it smartly. Make sure you're having a 3 to 4 year, maybe 5 year break between your pulse crops so that you're delaying the buildup of that inoculum in your soil. We see a lot of problems in fields that were lentil durum, lentil durum, lentil durum, because you're just, every time you put a lentil in there every other year, you're just increasing that inoculum. And so the other big thing is that frequency of susceptible pulse crops in your cropping history is really what drives the disease. The problem is that we scientists have not been able to come up with that magic number. I can't tell you, yes, you grow pulse crops 4 times, you're going to get disease. Some fields we see four cycles of pulse crops. Other fields we've seen 8 to 10 cycles of pulse crops. So it's really, really difficult to come up with that magic number. And because a lot of it actually has to do with what that soil moisture was the last time you grew a pea or lentil. So if every time you happened to grow a pea or lentil it was a really wet year, that's bad luck for you. If every time you grew pea or lentil it was a dry year, you're probably in better shape, although maybe your peas didn't do so well because it was dry. So I say luck, but there's probably some science behind that luck. We just haven't quite figured that out yet. So that's basically all I have to say about prevention. And now we're going to move into, okay, so you have root rot. Now what do you do? And really our only option right now is this avoidance, an extended break away from pea or lentil. And that's not what we want when we're talking about how important these crops are to our agriculture climate change solutions. So the next big question is how long? And I left a big blank space there because it's something that we don't know. And I'm really going to talk probably the next 5 to 6 slides is sort of working through, okay, how do we figure out how long do we have to maintain that break? And it really comes down to this. How many oospores to cause disease? Every time I put up the slide, I feel like it's a joke. I just don't know what the punchline is yet. So someone can come up with the punchline. That would really help out my presentation. And basically, you know, what we're looking at is, okay, well, what is that threshold level in the soil? Where do we have to start worrying that we've gone up to the next level? And so we've done a lot of work with this, some really ugly, sort of complicated models that I've been working with a statistician to try to figure out. And I just tried to make it simple and not show all these nonlinear regressions that he's done, and just break it down to this. Well, how much ooze spores do we have that causes 50% root loss of your roots? And basically, it depends on your soil type. In a black kind of loam soil, we found between 50 to 85 ooze spores per gram of soil. In a brown silt loam soil, about 60 to 100. And then in a dark brown soil, about 60 to 100. So really, we're looking at kind of an ooze spore threshold dose of about 50 to 100. That threshold is lowered. Again, we talk about our root rot robbers, our gang of root rot robbers. You add those in, it lowers that threshold dose. So it just makes the whole problem worse. And then some interesting data that we got out of Saskatchewan. I know that's kind of hard to see this picture, so you just kind of take my word for it, and I've summarized it in the side there. We went doing disease surveys, and my group collected roots and the group in Swift Current collected soil. We would test the roots to see how diseased they were. They tested the soil to see how many ooze spores they were. And in soil, in fields that were diseased, on average, there was about 1,000 ooze spores per gram of soil. So if we're talking about a threshold of 50 to 100, we can see that a lot of these fields are already well above threshold. Lentils was interesting because most of those fields were actually a little bit lower, at about 100 ooze spores per gram of soil. And that's generally because they are grown in the drier areas of Saskatchewan. And then what I thought was really interesting was that rangeland. I'll just break it down here. They looked at both kind of native and tame pastures. We can actually find very low levels of Aphanomyces, even in these fields that hadn't been cropped to peas or lentils before. Very low levels, kind of in tame pastures, looking at about, you know, what is that there? It's about 1 to 10 ooze spores per gram of soil, and the same with natives, about the same. So what that tells us is that there is a pathogen that's present in the soil, very, very low levels. Once you start introducing its favorite crop, those levels increase and you get above threshold. So say we get above threshold, then how long does it take us to get back down to threshold? And this is really the big unknown question. I came up with this sort of hypothetical ooze spore decay curve, and this was from a paper in the 80s, where they estimated it was about it took a year to lose 50% of your ooze spores. I don't believe that's correct. It was done a long time ago, so they were really just estimating. But if you started that red line, you're starting at about 1,000 ooze spores per gram, and you can see that it's going to take you about 6 to 8 years to get to below 100 ooze spores per gram. But we don't want to plant peas or lentils at 100 ooze spores per gram because you're just at threshold, you'll bring it back up, right? So it's going to take longer. So that's why we're talking about this sort of 6 to 8 year break. If you're just at threshold, maybe you're good with the 4 to 5 years. So that's why we say, to begin with, for prevention, practice that good rotation. If every time you grow up your lentil crop, you've increased your inoculum, wait 4 to 5 years, let it go down again, plant it again. We did just start a really exciting trial. If anyone follows me on Twitter, I think we posted about it. We are trying to get what is that specific number. So what we did was grow up roots. We stuck them in cages, little root jail for these root rot robbers. And then we went and buried them in the field, put them in these trenches, buried them it's about 10 inches deep, kind of about where you would expect kind of the root zone to be. Sorry, not 10 inches, 10 centimeters. And then they're buried, they're sitting there. We're going to dig out our first set, I think, next week. So we're just trying to see, okay, well, first of all, we're seeing if this crazy scientist thing the scientist dreamed up, whether it actually works. So we'll pull them out and see whether we can start seeing how much we're getting a reduction of those ooze spore levels in these roots over time. So yeah, we're pulling out our first set in February next week. So fingers crossed that we kind of see that this method works. And then we're going to just follow it for the next 3 to 4 years for as many of those little cages that we built. So hopefully some good results. And I've been talking to an economist to try to build some of these models for try to guide what your rotation, how long your rotation should be, and he says that he needs to know. This is the data that he needs to know to be able to start looking at those economic models. So hoping that this gives us some good data. And then the other thing that we're looking at, that just really exciting data that came off and really timely because of the issues we had this year, is how does soil moisture drive disease development? And so we look here, we looked at growing peas in soil that was either at low volumetric water content, moderate volumetric water content, or high. And low would be kind of simulating drought conditions. Moderate would be perfect conditions that we all hope for while we're growing stuff. And then high would be those really wet years, really like your soils never dry out in May, basically. And we looked at both Aphanomyces fusarium and then a combination of Aphanomyces and fusarium. And what we can see from those white bars is that Aphanomyces was really only a problem under those high, fully saturated soil conditions. Fusarium avanaceum was actually a problem across all our soil types, so it's not as dependent on soil moisture. What's really interesting and brings us back to this idea of this gang of root robbers is you put Aphanomyces and Fusarium avanaceum together, and we can see that disease severity was always much higher than either of them on their own. And even under drought conditions, we saw very high root rot levels. And that actually mirrors what we saw in 2021. Even though it was dry, some of our sites still had really high disease levels. And then when we look at, well, how much pathogen is left behind on those roots under both the low and the moderate, very, very low levels of pathogen. Look under that high, completely saturated condition. That's when all your Aphanomyces gets left behind on the roots. So that's why when we're talking about soil environment, if you're unlucky enough to always have your pea and lentils grown in a really wet year, that's why you're always returning so much inoculum to the soil. That basically just sums up what I said. So soil moisture is not as important for Fusarium root rot development. It was worse when both Aphanomyces and Fusarium are present. So all that to say, okay, well, how long are we staying out of pea and lentils? And really what it comes down to, and I think in my introduction it said, this is what I've been chasing the dream really for the past 10 years. Every time, sometimes scientists, I think we're really, really optimistic. Like I get so excited every time I do a new trial. And then you're like, oh, it didn't work. Let's try something else. And that's what scientists, we're eternally optimistic, I think. And so really what we're trying to do is take away that guesswork of how long do you need to have that break between pulse crops. We want to be able to go and test your soil and tell you, like, this is exactly what is in your soil and this is the quantity. It's been very difficult. Again, I think with COVID, we're all a little bit more literate in this idea of testing. So we have very high false negative rates from our soil. And this is because those ooze spores, they're really thick, the things that make them be able to withstand environmental conditions also make them be able to withstand kind of the tools that we have for testing them. So really hard to crack open the cell walls to get into the DNA. And the DNA is what we're testing with the PCR test. Everyone knows what a PCR test now is, right, too? So it just makes it challenging. So when it's dry, we get really, really high false negative rates. When the soil's wet, we actually do okay. But that means, well, we can only tell you if you have a risk in a wet year, but we know it's still there in a dry year. And then each soil type also has a different threshold, much harder to extract the DNA from clay soils because the material gets so tightly bound to the clay versus some of these black soils. So what can you do now if you know that you've had a root rot problem in the past and you're thinking about planting a pea or a lentil crop in 2022? And again, I think it's easy for me as a scientist to stand up here and say, well, you should have records for the past 10 years and you should know how all your pulse and lentil crops did 10 years ago. But I know that that's not always the reality. I guess what I would stand up here and say is start taking records so that as we move into the future, you can look back on these. But really, it's recognizing how your pulse crop did the last time you grew it. And that really will determine what your rotation interval is for the next 4 to 8 years. But I just want to caution that 2021 was a dry year. We did see root rot levels were fairly low, but like I've said before, that doesn't mean the pathogen's gone away. It's like it's sitting there laying in wait, waiting for those juicy roots and for a wet year. And so if we're reflecting back on how things did, I like to kind of put up this slide with different scenarios. So if your peas looked like this the last time you grew them, nice, healthy, green field, very hard to walk through, got a good yield, I would say, you've had your 3 to 4 year break, you're safe to plant peas or lentils again. If your fields look like that one down on the right-hand corner where you had big empty patches, really poor yield, don't plant peas or lentils in it in 2022. You could try a soil test to see if those levels are still high, but be cautious of those false negative results. And then this kind of scenario here on the left-hand side, those are the ones that are a little bit tricky. What we're seeing is you're starting to see little yellow patches. I don't know how well it shows up. You can see kind of yellow patches down in there. I think there's some, I can't quite see it, so there's some there, but usually along the water tracks or places of depression. Those ones are ones that you maybe have to think. You might be able to get away with planting a pea or lentil in there again, provided it's a dry year, not a wet year, but how do we predict that, right? But what can happen in a wet year is that those little patches can spread to the whole field in a really wet year. Whereas if it's in a dry year, they'll stay in those patches and it won't be a problem until the next time there's a wet year. But so what we do recommend for those ones is to soil test those fields, particularly those low spots or places that you saw were not yielding very well. We are hoping as part of Living Labs, if it gets approved, we are hoping to do a project with KSHAV looking at kind of remote sensing to be able to detect these kind of early yellow patches before it becomes a problem. So we can kind of drill down into, okay, where are the problem areas of the field for testing? And so soil sampling. So again, this idea of knowing our risk. The problem is right now is that we really are only as good as a present or an absence because our limit of detection is about 100 oospores per gram, just because of the nature of those oospores. So basically, if you get a positive result back, it's kind of telling you that you're already at threshold. We are working on trying to detect below threshold, but again, there's been a lot of challenges because of these oospores are just really, really frustrating to work with. And this graph just shows that really what we're looking at is that detection works well when you're already at a high-risk field. So here we looked at sort of our disease severity below 3. Those all came back as being negative, even though we knew that they were positive. And then once we get to these really high disease severities, it's when we can start to detect them. So just some cautions, I guess, with the soil testing. And so I'm just about done. We're looking at 2022 planning. What are we going to put into our fields for 2022? You can start soil sampling. I think I originally made the slide in the fall. I don't know if you can soil sample today when there's snow out there, but you can sample in the spring for fields that you're interested in putting peas in. We do recommend it to give you an idea of that field, but just beware of those false negatives. If your test comes back negative and you're like, no, I just know my pea crop did really bad last time, then trust your gut rather than what that test is telling you. And yes, sampling, where you sample is really important. Those low spots, areas where water tends to collect, like your fields, the landscape of your fields. I'd say we had really good success going into fields completely blind, just sampling in those low spots and coming back with a positive result. Okay, so just going to sort of end off bringing it back to what we talked about at the beginning, that when we know that pulses and legumes are really an essential component of sustainable agriculture systems, like Ken said, a really, really easy way to get kind of your 30% reduction of fertilizer use. So what can we grow in this kind of landscape of these root rot robbers? So pea and lentil, if you have a root rot problem, stay away from them. Soybean, faba bean, and chickpea are actually all really good options for some of these disease fields. Again, me as a scientist being able to stand up here and tell you that, but recognizing that they do not have as broad spread adaptability as our pea and lentil crops do, so they're better regional fits. Dry bean is okay. You're just talking to some of the Viterra guys. For now, it looks like dry beans, it's a different Aphanomyces in dry beans than it is in peas, so you're okay there. Alfalfa is, it's a yes-no. It's complicated. There's a race structure as well in Aphanomyces. Some races will infect some cultivars of alfalfa, not others. We don't do that testing in Canada. In the States, they're all kind of labeled as whether they're resistant to race 1 or race 2. Canada, we've never done the testing because we never really thought it was a problem here. So there's a bit of caution with alfalfa. Vetches, you know, if we're looking at these cover crop legume species mixes, vetches are extremely susceptible to Aphanomyces, so that is something to watch out for. Clovers, again, it's different species. Some are susceptible, some are not. Lupins appear like they're resistant. We just did some testing in 2021 and I haven't collected all the data yet. Okay, and so in a perfect world, this is what I think we need to move towards for this root rot management. Like I said, we're chasing this dream of having this reliable ooze spore quantification soil test because that's the foundation of any management strategy. We need to know what's in those fields. I mean, I guess it's the same as COVID, right? Test, test, test. That's how they thought we would solve it at first. So it's the same with this disease. Test, 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 and maybe we'll end up solving it. And then on top of that, we build more models. Okay, what's that interval period that we need? And let's start getting some partially resistant cultivars and some good agronomics in there for kind of our best management practice. So just end with, thanking. This is a big, big project. It's not just my inquest. This is an inquest from a lot of different scientists that are looking into this with a lot of different funders. And then besides my passion for Aphanomyces, my other passion is hiking. And I always like to say, climbing a mountain is just like research. Feels like you're never going to get there. And then once you do, it's always worth it. So with that, I can take questions.
Hi, do you do any work with irrigation at all, or is it just like the wet soil, or what do you?
Yeah, so we have seen, there's a lot of processing peas on irrigation, and we have seen that Aphanomyces is a problem on irrigated fields as well. Probably a little more so because they always have the moisture that's required. Yeah, so I don't know if that answers your question. Yeah.
Have you ever looked into intercropping for disease management? I've heard that's very helpful.
Yeah. Yes, and I didn't, I could probably can I give a 3-hour presentation next time? Yeah, we've done some work with intercropping, particularly with mustards, like some of the brassicas. What's interesting is that we haven't seen a mitigation of the root rot. The root rot severity is still the same, but we do get a yield bump in peas. So there's some interplay going on there, whatever it may be, that it does allow the peas to kind of tolerate root rot a little more. So we just finished a whole bunch of trials with more intercropping in 2021. Unfortunately, because it was so dry and we were on a dry land site, didn't get great results, but we had 2 years of data previous just looking with mustards and canola, and that is what we found.
Syama, what's the proper process to test the soil?
Yeah, so most of the soil testing lab. Oh, sorry, he was asking about sampling process for testing your soil. And a lot of the soil testing labs do offer the test, so like SGS offers it. I don't know if 2020 does yet. Maybe in Alberta it's only SGS. But they kind of have instructions on their website, but what I would recommend is sampling within that sort of 12 to 15 centimeter layer, really the top layer, because that's where the pea roots and lentil roots, they're pretty shallow, that's where they sit. So we recommend taking from that kind of top layer, kind of multiple samples within your area of depression, and then you can just bulk it together and send it to them. They don't need a lot, they usually need about 500 grams of soil. And that's another challenge with this test, is that you're looking at how many hectares of field, and then you're submitting 500 grams of soil. So the other thing we do recommend is if you can afford it, to take kind of multiple spots, and then it increases your chance of those reducing the false negatives. Oh, I see a question out of that.
In addition to that, does soil temperature make a big difference when it's taken?
No, so the question was whether soil temperature makes a big difference. Soil temperature isn't a big issue. You can sample in the fall, you can sample in the spring. Basically, whenever you can get into your field is okay to test. It's really soil moisture that is a little more important, but we can't control that, right? And something that we are just looking at is, okay, well, if we collecting dry soils and re-wet them in the lab and let them sit for like a month, is that gonna solve the problem? So that's something we're trying. We're also trying to like incubate them with some pea juice and seeing if that just wakes up the ooze spores enough that we can get them cracked open.
Well, certainly a fascinating area of study. Oh, don't go too far, just a second. A fascinating area of study and just such a creative way of making it interesting and one we won't forget. So thank you very much. Let's thank Dr. Syama Chaterton again, and we've got your custom shoe horn and a card. Thank you.